Hello, everyone. I'm going to be talking about our research work to test the bovine plasma hydrolysate capacity to increase iron concentration, transport it, and make it more bioavailable in a in vitro model of intestinal cells. This is our more recent article about the experimental work. Our group in Medellin, Colombia, is the Nutrition and Food Technology of University of Antioquia. However, 70% of this work was developed in the Bionutrients Research Group in University of Valencia, Spain. Anemia and in general, iron deficiency. Anemia have great prevalence worldwide. According to the WHO, women and children in school age are the ones that suffer the most from this pathology. There are many factors that contribute to iron deficiency, and the main reason is a diet with low iron bioavailability. The WHO and several institutions around the world have analyzed a strategy to fortify food that contains iron, especially in regions with a prevalent lack occurrence of good nutrition and iron. However, fortification of food with irons brings some problems such as a sensory change, sediment formation, lipid oxidation, among others. They determine the efficiency of this substance in enhancing to bioavailability of iron. The CACO tool cells line is the most used model to evaluate the absorption of iron at the intestinal level. CACO2 are human colon carcinoma cells suitable for studies of bioavailability of iron from foods. Enterocytes are epithelial cells of the intestine responsible for the absorption of various essential nutrients, the transport of water and electrolytes into the body, and the secretion of protein in the intestinal lumen. They have an abundant number of microbials in their apical pool, which increase the area for both digestion and intestinal transport. Enterocytes does the fundamental function of forming the intestinal barrier that presents biomechanical, biochemical, and immunological characteristics. Some studies show the iron interacts with different dietary compounds during digestion and for complexes that can increase the absorption. Ferritin is a protein in the immune systems responsible for storing all the iron in the blood systems. Ferritin is a protein specialized in the storage of iron in our body. It has the shape of a hollowed-out sphere or with an internal cavity, which constitutes the protein part called apoferritin, and which forms the covering that protects the iron from inside. In other words, the molecule without iron is called apoferritin, with a molecular weight of 413 mils to 418 mil daltons. And the molecule with the iron is called ferritin, and it has a molecular weight of 900 mil daltons. Its half-life is approximately 15 and 17.5 hours. It occurs in a high concentration in the liver, found spleen, bone marrow, and skeletal muscle. But it has also been identified in many other tissues in almost all cells in the body, including leukocytes, plasma, and even neoplastic tissues as a tumor marker. The methodology used in this work was the hydrolysis of the bovine plasma is carried out to obtain the hydrolysates. At the hydrolysates are determined the amount iron by atomic absorption spectrophotometry in order to know their content iron. After, the cytotoxicity in CACO2 cells and normal colon cells is determined by measuring the viability of the cells. With the hydrolysates, chemical tests to prove their ability to chelate iron and transport it. With the results at the best samples, the ability to make iron more bioavailable in CACO2 cells is evaluated. Then first, the cell viability is analyzed with the pre-treatment with the bovine plasma hydrolysate, and then the amount of ferritin formed in the cells is determined as an indicator of bioavailable iron at the cellular level. The results are obtained where, from the iron content determination tests by atomic absorption spectrometry, it was obtained that the test sample don't contain more than 5 ppm of iron, or parts of millions of iron, which is the detection limit of the analytical technique, which is good because that indicates the hydrolysate having additional iron added for a site, allowing for a more accurate measurement of iron in the experimental technique. The ability to chelate iron ions of the different degrees of the hydrolysis obtained was tested and the samples that gave the best results. It was the sample with a degree of hydrolysis of 19% is the best. That sample was fractionated to different sites of molecular weight, and its ability to chelate iron ions was tested too. It was obtained that the sample with the best capacity to chelate iron was the fraction less than 3 kilodaltons and greater than 1 kilodaltons. However, its biological capacity was half the chelating capacity of the hydrolysate. The best fractions was purified by HPLC and tested again. where a similar result was obtained, that it suggests a strong interaction of the different fractions in the hydrolysate to enhance their chelating capacity. From the MTT assays, the cells were cultured in plates under appropriate conditions. After 7 days of normal growth and development, they were pre-treated with the hydrolysate and the fractions the fraction with the highest chelating capacity at different concentration for 24 hours, and their variability was quantified for NTT. In that experiment, it was obtained that the hydrolysate at a concentrate of 0.25 milligrams by milliliters, and the fraction of the hydrolysate at a concentration of 0.1 milligrams for milliliters protect the cells up to a level of control set of cells without pre-treatment. In the test to determine the ability to make iron more bioavailable, the cells were cultured for 5 days under the appropriate conditions. Then a medium without iron was used as a maintenance medium for 2 days with the objective of make iron deficient in culture cells. Then these cultures were treated with the hydrolysate and the fraction, as well as with the iron complex with the hydrolysate and an iron complex with the fraction for an additional 2 hours, and the intracellular ferritin formation was determined as an indicator of the amount of the bioavailable iron into the cell or absorbed iron. The results obtained were both the hydrolysate and the fraction increase the cellular bioavailability of iron. The hydrolysate had a special ability for potentiating at to 5 times the bioavailability of iron compared to cells pre-treated only with the iron source. This result is very promising. By focusing these types of products to develop foods that can transport iron to the intestine, but also make it more bioavailable than this important micronutrient. The reference used in our work, and thank you for your attention. I hope you had understood my English. Thank you.